The cell cycle is an ordered set of events culminating in cell growth and division into two daughter cells. Non-dividing cells aren't considered to be in this cycle. There are 4 stages in the cycle: G1, S phase, G2, and mitosis. G1 stands for gap1, S stands for synthesis, and G2 stands for gap2. CDK, cyclin-dependent kinase, adds phosphate to proteins and are major control switches for the cell cycles, causing the cell to move from G1 to S to G2 into mitosis. MPF is a maturation promoting factor, which includes the CDK and the cyclins that trigger progression through the cell cycles. Mitosis is a nuclear division plus cytokinesis and produces 2 identical daughter cells during prophase, prometaphase, metaphase, anaphase, and telophase. The cell is engaged in metabolic activity and performing its preparation for mitosis. Chromosomes are not clearly discerned in the nucleus during interphase, but the cell may contain a pair of centrioles, which are organizational sites for microtubules. In prophase, the chromatin in the nucleus begins to condense and become visible in the light of microscopes as chromosomes. The nucleus disappears and the centrioles begin moving to opposite ends of the cell and fibers extend from the centromeres. Some fibers cross the cell to form the mitotic spindle. During prometaphase, the nuclear membrane dissolves, marking the beginning of prometaphase. Proteins attach to the centromeres, creating the kinetochores. Microtubes attach to the kinetochores, and the chromosomes begin to move. Okay, I know I just said prometaphase, and I skipped one. But during prophase, the chromatin in the nucleus begins to condense and become visible in the light of microscopes. Or did I just... Oh, crap, I already said that. Okay, never mind. Okay, awkward. Pretend that never happened. Okay, well, in the process of metaphase, spindle fibers align the chromosomes along to the middle of the cell nucleus. This line is referred to as the metaphase plate. This organization helps to ensure that in the next phase, when the chromosomes are separated, each new nucleus will receive 1 copy of each chromosome. During anaphase, the paired chromosomes separate at the kinetochores and move to opposite sides of the cell. Motion results from a combination of kinetochore movement along the spindle microtubules and through the physical interaction of polar microtubules. In telophase, chromatids arrive at opposite poles of the cell, and new membranes form around the daughter nuclei. The chromosomes disperse and are no longer visible under the light of the microscope. The spindle fibers disperse, and cytokinesis, or the partitioning of the cell, may also begin during this stage. In animal cells, cytokinesis results when a fiber ring composed of a protein called actin around the center of the cell contracts, pinching the cell into 2 daughter cells, each with 1 nucleus. In plant cells, the rigid walls requires that a cell plate to be synthesized between the 2 daughter cells. Diploid cells, which are 2N, have 2 complete chromosomes, and body cells are diploids. Haploid cells, which are just N, only have 1 complete set of chromosomes. Sperm and egg cells are referred to as haploids. Therefore, during mitosis, when the parent cell splits into 2 daughter cells, if the parent is diploid, the daughter cells will be diploid, and if the parent is haploid, the daughter cells will be haploid. This type of cell division, in this type of cell division, multicellular organisms grow and repair damaged tissues. The process of meiosis produces daughter cells with only half of the chromosomes as the parent cell. Meiosis enables organisms to reproduce sexually through gametes, which are sperm and egg cells that are also haploids. Meiosis is necessary in sexually reproducing organisms because the fusion of two gametes, which is also called fertilization, doubles the number of chromosomes. Meiosis also involves two divisions, producing a total number of 4 daughter cells. Thanks for watching!